This is Dr. Nicole Ross, joining us from UPenn. Dr. Ross received her bachelor from University of Idaho, and then she moved to New York to do her PhD with time option and aerosimoncelli, which is beautiful work on the special properties of V1 and MG neurons. I read those papers when they came out. It was beautiful. I highly recommend to people at IPM, our trainees, to read those papers. And also she wrote a very beautiful perspective in the praise of artifice with Tony Marsham, which I still go back and read it from time to time. After NYU, she moved to Jim DeCardinal's lab at MIT, and she applied a rigorous mathematical thinking to the ventral pathway, and she did very beautiful work with Jim. And since 2009, she moved to UPenn as an assistant professor, where she's now an associate professor there, and now she moves from another area of the planet brain to areas involved in memory, familiarity, and how we learn an object from just seeing it once. Dr. Ross received many awards. Just for one, I mentioned the Troland Award, which she received earlier this year for her fundamental contributions to the understanding of how the cortex makes use of complex visual information to guide intelligent behavior. It's an honor to host you today, Dr. Ross, and we are looking forward to your talk.
So thank you so much, Mendy. That's such a flattering introduction. I really appreciate that. Let me just get my slides going. Please let me know if there are any problems here. Pointer. Okay. Yeah, it's such a pleasure to join you. I'm really flattered by the invitation. I do hope that I'll be able to visit you in person sometime. I would love to come visit Iranian Neuroscience, but in the meantime, it's just really great to be here with you virtually. So today in my talk, I'm going to be talking about visual memory in the form of how we remember what we've seen, and particularly the images that we've seen. And what compels me to shift in this direction, and what compels the rest of my group also to work on this, is the remarkable ability that we all have to remember images. So it turns out that I can, for any of you, show you big stacks of images. And by that I mean thousands and thousands of images. And I can show them each to you, each only once, each for a few seconds. And afterwards I can give you a memory test. And you'll be really, really good at remembering the ones you've seen, and you'll be able to differentiate those from images that you haven't seen before. And so we've known about this remarkable form of single trial learning since the early 70s. I'll be talking a little bit about Lionel Standing's work. But what we still don't understand very well are the neural correlates, the mechanisms in our brain that support this remarkable form of learning. And so this is where my lab is trying to contribute. So we complement all of this beautiful work that's been going on in human behavior and human fMRI with investigations in the one animal model to date that's been demonstrated to recapitulate this remarkable human behavior, and that's the rhesus macaque monkey. And in rhesus monkeys, we can record at the exquisite spatial and temporal resolution that we believe is really important for unlocking the secrets of how the brain does this. So today in my talk, I'm going to be telling you about the work we've been doing in the past handful of years since my lab has shifted its direction to investigate these questions. But before I do, I just want to begin by telling you a little bit more about the behavior itself. So documentation of the behavior goes back to this classic paper by Lionel Standing, aptly titled Learning 10,000 Pictures. It's also investigated in more sophisticated ways this day in groups like Oliva's group at MIT. And these experiments themselves are pretty straightforward. They're just as I described. You bring a subject into the lab or online and you have them look at images. So each only once, each for a few seconds. And the key is they need to look at thousands and thousands of images, either over the course of hours or maybe even several days. And at the end of this exposure phase, you give them a memory test. You simply put two images side by side and you ask them, which one did you see? And so I'm going to be showing you the results from this study published by Tim Brady and colleagues, where subjects viewed 2,500 images over the course of 5 hours, again, each only once, each for a few seconds. And then they were given this, which one did you see memory test? And remarkably, subjects were over 90% correct at determining which of 2 images like this was embedded in their stack of 2,500 images that they saw on single trials. Even more remarkably, subjects were really good at picking apart which image was embedded in the stack when they were given 2 alternatives like this. So 2 very visually similar images. Notice it's the same bread, it's the same bread box, but here the bread is in the front of the bread box, and in this case it's in the back. And subjects were nearly as good, so 87% correct, at determining which one they had seen. So what this tells us is that we're storing these memories in our brains with remarkable visual detail. We're not simply storing memories of the objects that we've seen. We're storing a lot of detail about the specific configurations and the context that we saw those objects in. So just to summarize the behavior, this is an example of single trial learning. It happens at vast capacity, and it happens with remarkable visual specificity. And it's this behavior that we're trying to find the neural mechanism or understand how the brain supports it. So I want to emphasize that this type of memory is a little bit different than other forms of memory you might have heard of. It's often referred to as familiarity or recognition memory. To differentiate it, I like to evoke this experience that I think we've all had. So we're walking down the street and we see someone and we have that perceptive remembering. We have that aha moment. I remember you. I know you. And sometimes this perceptive remembering is even isolated from any ability to recall anything else. So we can't remember who they are or how we know them. We just know that they're familiar to us. So that's the type of memory we're investigating here. It's a binary decision. Have I seen you before? Yes or no? And today in my talk, I'll be telling you about our hunt in the brain for what I'll call the memory signal. So insofar as you're walking down the street and you have that aha moment, that sense that you've seen somebody before, there must be a pattern of neural activity that's driving your perceptive remembering. And so we're interested in figuring out where is that signal in the brain and how does it manifest. And this signal is really important to us because I think it's really the key, the bottleneck to unlocking everything about how visual memory works. So if we can't figure out what that signal is, we can't figure out much. Or if we can figure out what that signal is, then understanding how visual memory works is really the question of how does that signal come to be. So that will be part 2 of my talk. But before I present part 2, I'll be telling you about a phenomenon known as image memorability. It's a signature of visual memory. It simply refers to the fact that some images are easier to remember than others. So it's systematic variation in memory that we all share. And I'll tell you about the correlates of memorability in both the brain as well as the deep neural networks that are trained to categorize objects. And it turns out that the correlate we've discovered for image memorability really flies in the face. It is at odds with our current proposals about what the visual memory signal is. So part 2 is really going to be a description of why the correlate of memorability is such a problem for theories about memory, and then a resolution of that problem as well. Okay, so I'll just jump right in here with image memorability. So image memorability, again, like I said, it just refers to the fact that some images are easier to remember than others. And so here I'm showing you on the slide a bunch of different images configured by their memorability. So up here in the upper left, we have the most memorable images, and then we go down here to the lower right, where we have the least memorable images. And these are taken from this nice paper by Khosla et al. It turns out that this memorability variation, it's very systematic. So a large fraction of it is shared between us. So we all tend to find the same things memorable and forgettable. And what that means is we can quantify memorability for individual images. That in turn allows us to do things like configure memorability on this slide. But more importantly for this talk, to actually investigate the correlates of memorability in the brain, because memorability is a property of an image that we can investigate. We can ask the question, what differs in the brain in the representation of a highly memorable image versus one that's easier to forget? So the way we score the memorability of an individual image follows from the introduction, again, from Oliva's group, and this nice paper by Philip Isola, and a companion paper by Wilma Bainbridge. And this is in the context of a recognition memory task. So subjects are looking at individual images on each trial, and they're asked the question, have you seen this image before, yes or no? And so they'll see images in a sequence. Embedded in the sequence are these short repeats, so these repeats after a short delay, those are called vigilance repeats. They're just there to make sure that the subjects are paying attention. And then after these longer delays, so after hundreds of trials have elapsed, after minutes have passed, there'll be a repeat of something that was shown earlier. And that's where memorability is scored. Memorability is simply the fraction of subjects that remember seeing an image that was presented minutes ago, and it's scored from 0 to 1. So 1 indicates an image that everybody will remember seeing. 0 is an image that people will remember seeing a chance. And if you look at large databases, such as the 60,000 photographs chosen from random sources that comprise LaMem, you see a really big variation in memorability across images. So there are some images that will get scores near 1, meaning that nearly everybody remembers seeing them. And then there are some images that will get scores less than 0.5, meaning that there are images that the majority of people will forget. And the mean amount of remembering is 76%. So there's a variation here that needs to be accounted for, and it's that variation we're interested in explaining. Importantly, these scores are reliable and predictive. So if you measure memorability scores for images in one group of subjects and you ask how well do they predict how other groups of subjects will remember those images, the rank correlations are really high, meaning that we're all sharing this component of memorability. So we all tend to find the same things memorable and forgettable. It's not that all of the variation can be explained by shared variability, but the majority of it can. Okay. So memorability is really interesting and fascinating, and if you want to learn more about it, I encourage you to have a look at this review that we wrote recently. In terms of the mapping of image content to image memorability, I won't talk a lot about that in this talk today, because here I want to focus on the neural correlates, but I'll just give you a summary. We know it's predictable insofar as Ulova's group has developed a deep neural network that can take any image and produce a memorability score prediction for it. And that score is a really good prediction. So it's a really good prediction of how memorable human subjects will find that image. So it is predictable. At the same time, what's fascinating is it's completely counterintuitive what it is. So if you ask naive subjects to predict how memorable images will be, there is zero correlation between their predictions and how memorable images actually are. They make predictions based on things like how beautiful the image is, things like that. But that is not what drives memorability. In terms of what does drive memorability, it's highly multifaceted. So there have been about, I think, on the order of 70 different attributes that contribute to memorability variation. For example, we know that images that contain faces tend to be highly memorable, whereas images with nature scenes tend to be unmemorable. Images with atypical content tend to be memorable, whereas images with typical content tend to be less memorable, and so on. So it's really highly multifaceted. To date, no one has found a single principle that drives memorability variation. So I won't say anything more about that. But rather, we're going to start in this talk, I'll just start with the groundwork. There is this variation. And so how is it reflected in the brain? That's the idea. Okay. So the work I'm going to be talking about was a really fun collaboration between my group and the group of Ulova's, and it was led by, it was kind of a side project led by a number of postdocs, including Vahid Marpour, Drew Jagel, and Travis Meyer from my group, and Yalda Mosinzada from Ud's group. And we started the project knowing where to look for the correlates of memorability, and that was in high-level visual cortex. And this was based on some nice work that Wilma Bainbridge had done using human fMRI, where she localized memorability to high-level visual cortex by determining that memorability could be decoded, using a linear decoder there, in classes of images. So, for example, memorable versus forgettable faces or houses and so on. And so this high-level visual cortex, it is the analog of what I'll be referring to today in my talk as monkey inferotemporal cortex IT. So IT in a monkey lies in the temporal lobe. It's considered to be the highest stage of visual processing in the ventral visual or form processing pathway. After signals leave IT, they go into multimodal association cortex. And IT has been implicated extensively in higher level visual computations, including object recognition. So our hypotheses going into this project about how memorability might be reflected there followed on, they built on the framework that's been established for thinking about how objects are represented in IT. So I'll just describe that to you very quickly, and then I'll explain to you how we built on it. So when we're thinking about how identity representations are reflected in IT, so the difference of an image of an apple versus a flower, we think about that as what I like to refer to as a pattern of spike representation. Meaning if you show an apple image to IT, you get a particular pattern of spikes across all of the neurons in the population. And if you show a flower, you get a different pattern. This very simply refers to the fact that some neurons in IT are apple preferring and some are flower preferring, and so you get these different patterns. And we tend to like to think about these representations in these high dimensional spaces where the dimensionality is defined by the number of neurons in the population. So in IT cortex, we're working in a 16 million dimensional space. But we can intuitively think about what's happening in that space in just 2 dimensions. So the vigor of the response of unit 1 versus the vigor of the response of unit 2. And these different patterns of spikes across the population, they simply amount to different vectors that are pointed at different positions in this high dimensional space. And that's how we think of identity being represented in IT cortex. You get clusters of all of the representations for a particular object like apple, and those clusters are separated from other clusters for representations, for example, say flower. So our first hypothesis about memorability was that it might also be reflected in a pattern of spikes representation. And one instantiation of that might be an interference hypothesis. The idea that the most memorable images might be isolated out in their own part of the representational space, whereas less memorable images might be more closely packed. And it might be the case because of this close packing, you get interference in the transformation from vision into visual memory. So that's one hypothesis. Another hypothesis evokes a complementary coding scheme, and that is population response magnitude as opposed to pattern. And this is the hypothesis that the length of these population response vectors changes. You get different total numbers of spikes coming out of IT for more memorable as compared to less memorable images. And this is the simple hypothesis that if you start with more robust visual representations, that's going to lead to a better memory downstream. So these were our 2 hypotheses going into this study. I have to say that neither of the hypotheses at the time seemed very likely for different reasons. In terms of the first hypothesis, this seems inconsistent with what we know about visual memory behavior, and that is it's very visually specific. We can remember if we've seen the bread in the front versus the back of the bread box. And what that suggests is there's not a lot of interference between visually similar representations and the transformation from vision to visual memory. At the same time, this one also seemed unlikely because it was inconsistent with a lot of prior work that suggests that IT does not do this, including my own work when I was a postdoc working with Jim DiCarlo. I spent a lot of time looking at the representations for different images and objects in IT and comparing them with the inputs from before. And we found that every image in IT cortex basically evokes the total number of spikes, same total number of spikes. And this is consistent with ideas of homeosynaptic plasticity and divisive normalization, all of these active mechanisms that are really trying hard to maintain the total number of spikes across the population and just repatterning them. So again, neither seemed very likely, but we decided to forge ahead anyway. And so I'll just give away the punchline at this point and tell you that it's this one. So we have very strong evidence that this is implicated, and we have no evidence in support of the alternative but not mutually exclusive hypothesis. So just to show you the evidence, so we collected data from IT cortex while monkeys were performing a task that was very similar to the one that's used in humans to score memorability. So on each trial, the monkeys look at a particular image and they make a behavioral report about whether that is novel or familiar by an eye movement response to 1 or 2 targets on the screen. So they'll see images early in the sequence, they'll be repeated later. For this part of the talk, I'll be focusing on the novel presentations of the images. In the second part of the talk, I'll be focusing on both novel and familiar presentations. But the question here is what differs in the representation of a memorable versus a non-memorable image the first time you see it. And we found that when we looked at the grand mean firing rate response across the IT population, it was strongly correlated with image memorability scores. So these are human-based memorability scores, but it turns out we also demonstrated that humans and monkeys tend to find the same images memorable and forgettable. So we can use this as a proxy, given that we can't record memorability scores from 80 monkeys to get them. So human-based memorability scores plotted against the vigor of the IT response. This is population response magnitude. It's the L2 norm of the population vector. You can just think about it as vigor. And you can see there's a strong and highly significant correlation. So each dot on this plot is an individual image. The Pearson correlation is 0.68. Okay, so we have this plot of population response magnitude versus image memorability. And because magnitude is not a very intuitive quantity, I've taken 2 points on the plot and I've converted them to just grand mean firing rates, which are much more intuitive. So this is the grand mean firing rate across all units in the population. And we see a highly memorable image, evokes a 15.5 spikes per second response, and that decreases by about 20% to 12.5 spikes per second for a less memorable image. So this is a non-trivial effect. Let me just remind you, this is the grand mean firing rate response across all units. This isn't about tuning, this is about grand means. Okay. So this suggests that there is at least some correlate of memorability, which is reflected in this way in population response magnitude. I'll also mention that we've looked for the alternative hypothesis, that there might be something else reflected in patterns, and to date we haven't found any evidence for that couldn't be accounted for by this population magnitude coding scheme. So what this suggests to us is that there is magnitude coding in IT. We didn't know that before, we know now. It exists across the class of natural images, such as photographs, and it has apparent behavioral consequences, namely for remembering what we've seen. It's accounting for this big memorability variation. We systematically remember some things better than others. In terms of just almost a side note of why we missed this before, I'll just mention that this memorability variation, it emerges pretty late in the stimulus-evoked response, about 220 milliseconds after stimulus onset. And we may have missed it because when you study object representations, you typically study them in this fast paradigm because object perception is so fast. So you'll flash images up for 100 milliseconds each. But this signal doesn't emerge until you have images on for a longer amount of time. And when we study memory, for good reason, we study it across longer periods of time. So after subjects view images for hundreds of milliseconds. Because in that fast paradigm, you can see, but you can't remember anything. It takes hundreds of milliseconds to store memory. And so this is probably why we missed this. But now we know it's there. Okay, so before I move on to the next part of the talk, I want to tell you about another follow-up component of this result where we investigated the origin of this image memorability variation. So this seems to be just variation in visual processing. And so we asked the question, does this type of variation emerge when you wire up a system to see and recognize objects? Or does it require that the system also remembers what it's seeing? And we were able to ask these questions, of course, because the field has these wonderful, deep artificial neural networks that are trained to categorize objects. So this work follows on this really beautiful work that's been going on in the field of object recognition, led by Dan Yammans and Jim DiCarlo and Nico Scorte and many others, that have demonstrated if you take a deep artificial neural network and you train it to categorize, recognize objects, what you get is a system that has a remarkable resemblance to the primate brain and how visual representations are organized there. So these deep nets have different layers, and it turns out that the different layers map on to different stages of the form processing pathway. So the earlier layers map on to V1, and then V2, V4, IT, and so on. And so what we're asking here is the question, so those systems were optimized to see, but they were not optimized to remember, and they can't actually remember anything. So once they've been trained and their weights are frozen, you can show them something once, you can show it something 1,000 times, they'll have no trace of that memory of what they've seen. And so the question is, do these networks also have memorability variation? Is it the case that in a particular layer, those networks will respond, that layer will respond more vigorously to some images over others? And is the vigor of that response predictive of what we humans will find memorable? That's the question. So to show you the results, I'll just remind you that in the actual IT cortex, we can summarize the results in terms of a Pearson correlation, which is the correlation between these human behavior scores and the vigor of the response in IT cortex. It's a single number. And we can plot that number across different layers of VGG16, as well as show you, but what I'm about to tell you is true of every deep net we tested, AlexNet, and so on. And so what we find is that the earliest layers, these correlations between population response magnitude and image memorability are small. And this is consistent with some of what we know about memorability, that it's not driven by low-level variation, things like contrast or luminance. It's a property that really belongs to something more like objects or higher-level representations. As you go to higher stages of the network, though, what you see is memorability emerge. And I think this is really remarkable. Again, what this means is that these deep nets are trained to categorize objects. Their higher layers respond more vigorously to some images over others. And how vigorously that network layer is responding is going to predict how well we are going to remember something. It's pretty remarkable. And at the highest stages, what you see is this drop-off. This is not surprising. These are the categorical layers where you get the grandmother-like cells, right? The ones that tell you what object identity you're looking at. They're there to group. Okay. So what this tells us is that image memorability is really, to a first pass, about vision. Not only can you see it in the responses to novel images in the brain, but it's also reflected in these deep artificial neural networks that are configured just to categorize objects, but they're not optimized in any way to remember, and they can't remember anything. Okay, so that's the first part of my talk. In terms of the second part of my talk, I want to focus on this memory signal, and I'll come back to the issue of how that first part challenges the second part. But first, let me just remind you that what we're trying to do here is we're trying to hunt around in the brain and figure out where's that aha signal, the one that tells you, I found what I'm looking for. And not sorry, I remember seeing you before. Where is that? So I'll just begin by telling you what we know. And laying out a framework which accurately depicts that it's complicated. So we know we should be looking in, not surprise, structures in the medial temporal lobe complex, places like perirenal cortex, entorhinal cortex, the hippocampus, and we should be looking at the inputs that are arriving to the medial temporal lobe from the ventral visual form processing pathway, places like IT. Now, in terms of what these structures are doing, we have good reason to think that perirenal cortex is probably the structure that's supporting the most remarkable aspects of this behavior insofar as our ability to remember vast numbers of images on single trials. This is a structure that nobody's barely heard of, and it's a really important structure for this behavior. If you lesion it, as shown by Beth Buffalo, you ablate familiarity behavioral reports. At the same time, it's not the only structure involved, so we also have reason to think that the hippocampus is important, particularly for the visual specificity of visual memory. So human patients who have natural lesions to their hippocampus as a consequence of something like a stroke, they are unable to have that same visually specific behavior, so they tend to overgeneralize. So maybe they'll see the bread in the back of the bread box, and if you ask them, have you seen the bread in the front of the bread box, they'll tell you yes, whereas control subjects will tell you no. Okay, so the hippocampus seems to be really important for shaping that visual specificity, and it's thought to do so via a computation known as pattern separation. It's also the case that this pathway is not purely feed-forward. So there's good reason to think that these signals, these memory signals, are probably fed back from the medial lobe back to IT. For example, if you look again at patients, case studies of human patients that have medial temporal lobe damage, you see that they have blunted visual memory signals back in their high-level visual cortex. So this pathway is really complicated. This pursuit is really complicated. We have many different brain areas involved. We have information going forward, information going back. This is the same story we find in all of, I would say, all of cognitive neuroscience, right? How are we going to tease all of this apart? And so it's the complexity of this pathway which really motivates me to want to pin down the visual memory signal. So to ask the question, in these brain areas, what is the pattern of neural activity that aligns with behavior? I think that that's really the key, the bottleneck to unlocking the question, how are these pathways interacting with one another? Where are the signals coming from? Where are they going to? We have to first identify the signals. So that's our deep interest in trying to do this. So in terms of what the hypotheses are about that signal, there's been one that's been around for quite some time, since the early 1990s. And that is that the signal driving that perceptive familiarity is what's called repetition suppression, both in IT cortex and then throughout the medial temporal lobe. And so repetition suppression, its existence is not in dispute. Everybody agrees repetition suppression is there. The real questions are, well, is this the signal that drives behavior, or is it an epiphenomenon? So repetition suppression simply refers to the fact that if you show a particular image to the visual system, you'll get a vigorous response. And then if you repeat it, you'll get a reduction in the response. That is repetition suppression. And the reason that repetition suppression has been implicated in visual familiarity behavior is because it has a lot of the characteristics that mimic or reflect the behavior. For example, repetition suppression remains visually specific even after seeing very large numbers of images. So it doesn't transfer across images that are different. So you still get a vigorous response to an apple even if you've seen a flower. And to get repetition suppression of the apple, you have to repeat the apple. It's also the case that repetition suppression lasts an incredibly long time. So as demonstrated by Zhang and Brown in 1998, they reported that repetition suppression after seeing an image only once can last up to 24 hours in the medial temporal lobe. So that's a single trial phenomenon that seems to align well with what we know about the remarkable aspects of this behavior. It's also the case that it's pretty easy to think about how the brain might decode familiarity from using repetition suppression. It implies something like a class of what we'll call the repetition suppression decoders. In the simplest instantiation, which is really good conceptually, the brain is simply counting the total numbers of spikes coming out of a brain area like IT. And if that exceeds some threshold, the brain calls that image novel. And if it's below that threshold, the brain calls it familiar. Okay? That's one of a class of decoders that are all about magnitude decoding in the brain. And so given all of this evidence and given the lack of any good alternative hypothesis, the first study we did some years ago now investigated whether or not this repetition suppression signal was quantitatively aligned with familiarity behavior. So here we looked at rates of remembering and forgetting. The idea is that monkeys, like ourselves, they tend to remember better the things they've seen most recently, and they forget things as a function of time over minutes. So you can think about the behavior as a plot of remembering as a function of time over minutes. You can configure the experiment, so monkeys will remember for long periods of time or short periods of time. Here it was minutes, long time scales in our experiment. And so this falls off, and the question is this fall off well aligned with the disappearance of repetition suppression in IT cortex? And we asked this question by looking at a class of linear decoders of a repetition suppression family, and we found that a subset of that class were indeed nicely well aligned. So the idea is you apply these decoders to the IT population, and you make a prediction of what the behavioral will be, and then you compare that to the actual behavior, and a subset of these repetition suppression classifiers are nicely aligned, not only with the choice, but also the reaction times of the monkeys. And this subset wasn't as silly as counting the total numbers of spikes coming out of IT cortex. That didn't really work. But it was the types of things that worked were things like optimized weighted linear decoding of the type that's used be positive, so neurons that responded more robustly to novel as compared to familiar images. Or another one that was very effective was counting the total numbers of spikes coming out of the 50% of most sensitive IT neurons. So the idea that not every neuron in IT is involved in familiarity, but the top 50% of neurons are, so a highly distributed signal. So that suggested to us that, yes, we're on the right direction with repetition suppression. It is likely to be the visual memory signal. But there's a big elephant in the room with regard to this hypothesis, and it's highlighted in the first part of my talk. and that is that memory is not the only thing that modulates the overall vigor of the response of IT cortex. There are also many other factors, namely memorability, right? The one I just described to you. And memorability turns out to go in the wrong direction to be consistent with the hypothesis that repetition suppression drives the behavior. So let me just explain that statement, break it down for you. So in terms of memorability behavior for an individual subject, we can just think about it like this. So different images have different memorability scores. And so an individual subject will do better at reporting or remembering an image as a function of those memorability scores. That's just kind of a restatement of the behavior. So you can think about that as the behavior of an individual subject. It turns out that the magnitude variation in IT cortex that corresponds to memorability, so more vigorous responses for highly memorable images than images with low memorability, introduces ambiguity. Meaning you can have cases in which you have familiar images that are highly memorable, that have stronger, more vigorous responses than images that are novel that have low memorability. And it turns out that this, because of the memorability variation, what happens is the prediction is that of a total spike count coding scheme, or the class of repetition suppression decoders, the prediction is that the most memorable images are going to be the ones that are actually the least memorable. So whereas this is the actual behavioral pattern of a subject, the class of repetition suppression decoders do this. And you can see that is a really great example of a line that does not go through points, right? It's got the wrong slope. So the correlates of memorability challenge the proposal that repetition suppression is the memory signal. It just cannot be right. And the question is, okay, well, if it's not that, then what is it? And so that's what I want to address in the next part of my talk. And so apologies for the bait and switch, but we're going to switch away from talking about memorability to talking about another variable that we are actually studying, and that is image contrast. And the switch to from memorability to contrast just has to do with the historical fact that we were doing these two studies I'm telling you about at the same time. So at the same time as we were discovering the neural correlates of variability, we're also deep into investigating this contrast modulation. But the story is the same with contrast as it was with memorability. The idea here is that high contrast images are going to have more vigorous responses than low contrast images. That's going to induce ambiguity. And presumably, we don't confuse contrast and memory. So we're not more likely to call novel images familiar. We're not more likely to call low contrast images novel, nor are we likely to call high contrast images familiar. So yeah, you get the sign. Okay. So how does the brain disambiguate contrast and familiarity? So this section of the talk was led by Vahid Mirpur, also with contributions from Travis Meyer in my group, and this is a collaboration with Eros Simoncelli at NYU. So the monkeys were performing a task which was similar to the task that I described before. So on each trial, the monkeys are looking at a single image and making a behavioral report. Is this image novel or familiar? The difference now is that monkeys are looking at the images in grayscale, and they're presented at one of two contrasts, right? They're presented at high contrast or low contrast. And we trained the monkeys to report that an image presented at a different contrast was in fact familiar, such as this dog here. So the monkeys are doing contrast invariant familiarity detection. And in this experiment, we had 6 different conditions. So we had, in the case of novel images, they were either presented at high contrast or low contrast. And then we had 4 familiar conditions, 2 of which were the same contrast. So novel repeated high, novel low, and then we might mix that. We also had 2 conditions where we mix the contrast. So high-low versus low-high. And we were successful at training the monkeys to disambiguate changes in contrast from changes in memory. So here's their behavior. So here I'm plotting just the proportion of times monkeys reported familiar for the 4 different familiar conditions and then the 2 different novel conditions. And so monkeys report that familiar images are familiar at a higher rate than they report that novel images are familiar. You can think about these as false alarms. And up here, I'm showing you the benchmarks for two different invariance conditions. So this is what we would expect the behavior to look like if the monkeys were perfectly contrast invariant. This is what we would expect the behavior to look like if the monkeys systematically confused memory and contrast. So they would report that low contrast images are familiar at a higher rate, and they would report that high contrast images are novel at a lower rate. So you can see that the monkeys' behavioral patterns look more like this than they do like this. For example, if we look at the two same contrast conditions, these two here, monkeys at the same rate report that high contrast familiar images and low contrast familiar images are familiar. And we can quantify that by just setting these benchmarks. This is the benchmark of 1, this is 0 invariance, and the monkeys had an invariance that was quantified at 0.9. So the monkeys are not confusing contrast and familiarity. In terms of the repetition suppression decoding scheme, we found that because IT is modulated by both contrast and familiarity, that is indeed confused. Stated very directly, the same decoders that were working for us in 2018 aren't working for us anymore under the challenge of contrast variation. And you can see that the pattern they take on, as illustrated in these bars, is exactly the pattern you would expect from contrast confusions. So repetition suppression cannot be the signal that's driving these monkeys' behavior reports of whether or not these images are novel and familiar in this task. So that's broken. The question is, well, what is the fix? And we hypothesized, just based on all of the evidence and support of repetition suppression, not only our own, but in many different publications, that it's something like repetition suppression, but it needs an adjustment. And we call the adjustment sensory reference suppression. This is the idea that what the brain is doing is it's still looking at repetition suppression, but it's correcting for that sensory-induced ambiguity for changes in firing rate by contrast. What we were happy to learn is that this correction can be applied not by accessing the firing rate for contrast modulations from some other brain area, maybe that's not modulated by familiarity, but it can be applied within IT cortex itself. So let me show you that evidence. So I'll call this SRS, sensory reference suppression. So here I'm largely plotting things you have already seen, but in a slightly different way. So over here, I just have sorry, I've lost the title here, it didn't show up, but this is just the repetition suppression or total spike count decoding scheme. That's what this is. And it's the one that doesn't work, right? So this is the monkey behavior and the dots. The bars are the total spike count prediction. This decoder systematically confuses changes in familiarity and changes in contrast. And here I'm showing you where that decoder falls in a 2D space. I've been showing you lots of 2D spaces. This is not the output of 2 neurons, but it's 2 dimensions of this high dimensional space, of this 800 dimensional space that we are working in, because that's how many neurons we have in our population. And along the diagonal, I'm showing you the total spike count coding scheme. Okay, so we'll call this is the simplest RS decoder. So these ovals, the gray ovals are correspond to probability distributions for the novel images, and the colored ovals for the repeated responses for familiar images. Okay, and so by projecting the information along this total spike count decoding scheme and applying a threshold, that's shown over here. And this is the problem, right? That you get dispersion within the class of novel images and within the class of familiar images. That's this variability in the predicted response here. Okay, so that's the thing that's not working. So sensory reference suppression suggests that we should start here, but what we want to do is correct for this contrast-induced variability. And so we can look to see where on this 2D plane information about contrast falls, and an optimized contrast decoder falls here. It is not orthogonal to the total spike count coding scheme. It can't be. We know that contrast induces changes in the total numbers of spikes. But it's actually quite far away from it. This is about 70 degrees away. And so what sensory reference suppression does is it says, take this information along this axis about contrast and use it to correct the total spike count coding scheme, which amounts to just shifting that decoder 20 degrees in the other direction. And now we have a new linear decoding axis, which we call SRS, in which, if we project along that, we've eliminated that variation within the class of novel images within the class of familiar images. We've made the decoder invariant. And now we can compare that decoder with what the monkeys are doing, and we can see it provides a much better prediction of the monkey's behavior. So that's what's shown in these bars. Like the monkeys, this decoder is by design invariant. So an SRS decoder that corrects the overall firing rate for changes in image contrast provides a much better account of behavior than does repetition suppression. And I want to emphasize that this correction that's required is available within IT Cortex itself. We did not have to access information to do the correction from some other brain area, but rather we could do all of this by applying a single linear decoding scheme to IT Cortex. And so this is our now working hypothesis about what the signal or the neural activity pattern that is used to drive behavior. It's SRS. That's our hypothesis. So one thing to emphasize is that this hypothesis does not just apply to contrast, but in principle, it should apply to other factors, including memorability. So let me just walk you through that conceptually. So memorability, like contrast, results from an ambiguation between along the total spike count coding scheme, because that is modulated both by memory or familiarity. It's also modulated by memorability, this variation you see across the different images. And so because of that ambiguity, the fact that both things are happening along this axis, if you try to decode along this axis, what you get is a misalignment between the monkey's actual behavioral patterns and the predictions of a total spike count decoding scheme. However, just like we did for contrast, as long as memorability lies somewhere else, the memorability decoder information lies somewhere else other than the total spike count coding scheme, we can orthogonalize to it. And in the case of memorability, we're interested in not just orthogonalizing, which would tilt this from a negative slope to a flat line, but actually over-orthogonalizing, so you get that positive slope. That would be the SRS decoder for memorability in principle. In practice, we're still trying to work this out. So this is now our new hypothesis that this should work. So we discovered SRS for contrast. Our hypothesis is that this should also work for memorability. We're currently testing that hypothesis, and the devil's in the details, and now we're just working on the technical challenges, things associated with the fact that you have to do a linear regression, for example, to find this axis, so you need more data, and so we're going to collect that data, and so on. But that is SRS. Okay. So just to wrap up, summarize, just want to leave you with two salient points. We started by asking the question, how do we remember the images that we've seen? And I've tried to make a strong case that understanding the answer to that question requires us to pinpoint the neural activity pattern that drives visual memory behavior, that aligns with visual memory behavior. The first thing we investigated was the question of why some images are better remembered than others. And we found that that was strongly correlated with the larger magnitude responses from high-level visual cortex, which was also reflected in convolutional neural networks that were trained for object categorization. And we found that this result challenged our current hypothesis about this thing we think is so important as the identity of the visual memory signal. And so what neural signal drives visual familiarity behavior? Well, in terms of the hypothesis space, we've at least ruled one thing out. It's not repetition suppression like we thought. But we found that a modified account of that may actually work, and that's SRS. And so now that's our working hypothesis that we're going on to test. And so finally, I just want to thank the funding sponsors that have been so generous in supporting our work. And looking forward to talking to you guys about these issues. So yeah, thank you.
Thank you so much. Great talk, very clear. Time for a few questions. Matthew, you can ask the question.
Hi, can you hear me?
I can. Hey, Matthew.
Hi, Nicole. Thank you. That's a very beautiful talk. I really loved it. And my question is concerns a line of research from Elizabeth Buffalo about oscillations and synchrony in oscillations, and the finding that stimuli that are better recorded are those that are processed when the pathway into the hippocampus is more synchronized at different levels. And I wonder whether the images that you find are more memorable images might be ones that induce more synchrony along the pathway.
Yeah, no, it's a great question. I don't know the answer to that. We now have electrodes in the hippocampus, and we're investigating how memorability changes across different levels of the pathway. And I think that there's a lot to be said. We could go on a deep dive for another hour about synchrony and how synchrony might be reflected in terms of the types of stories I've been telling you about how familiarity is reflected. I think the thing I would like to emphasize the most is that the signal, there's an important distinction to be made between where you see the signal versus where the memories are stored. And I think there's convincing and mounting evidence that these memories are stored in the medial temporal lobe and then they're fed back to IT cortex. And where they're actually decoded from, I think that's the question that's hot on my mind right now. But I think that the things that Beth was recording in the hippocampus are likely to reflect signatures of visual memory storage.
Okay.
And that there's some component of repetition suppression like stuff that is actually reflecting what's happening in IT. Some of it is feed forward from earlier in the pathway. I mean, the retina has some repetition suppression. It's very small and it's on very short time scales, but it's there, right? And then you get these cumulative feed forward effects. You get stuff in IT cortex for current connectivity, I think is involved. And then you get feedback from higher stages like the medial temporal lobe. So I don't think there's a single locus for repetition suppression. I think different components of the signatures are going to be contributed to in different ways, and Beth's work has spoken to, I think, an important component of the storage. Whether IT oscillates, I should know the answer to that question, right? We haven't actually investigated that particular question, but it's a good point. We should dig in.
Okay. Thank you very much.
Great. Ali, you have a question? Yeah, go ahead.
Hi, Nicole. It was a very good talk, as usual. I have a quick question. So you showed a scatter plot. X-axis was the memorability, y-axis was the firing of the IT neurons. I'm wondering, so given the fact that the IT is basically very sensitive to familiarity, novelty of the objects, I'm wondering if you were to regress out the familiarity of the objects, could you do then like a partial regression on the residuals of that, and to see if still memorability is encoded independent of familiarity of the objects? Because, like as you mentioned, intuitively, it's possible that objects that are more bizarre are more memorable, even though you said there's a zero correlation at some point, but still, it feels very, very strongly. I feel very strongly that there may be some correlation. So have people looked at that?
Well, thank you for the compliment, Ali. I appreciate it. So my description of what's going on there, and I'm sorry I don't have it as a back pocket slide, is that in the case of novel images, you get a certain regression, right? In the case of familiar images, you get repetition suppression, and the repetition suppression line has a slightly shallower slope, which is consistent with the hypothesis that repetition suppression is multiplicative, right? So 10% of 10 is a smaller number than 10% of 100, right? So what you're going to get is a multiplicative rescaling of firing rate. That's a good depiction of how across the population we get a multiplexing of memorability with repetition suppression. But I think that might not be answering your question. I mean, one of the things I want to emphasize is that I think that atypicality is in there. if I said something to mislead you to think that that's not the case, I don't want that. Let's correct that. So all of those, my not wanting to talk too much about what components of an image, what kind of content maps onto memorability. I think all of that content just like it maps right onto this firing rate axis, right? So I think IT is going to be this locus in which all of that variation gets kind of compiled together. Does that make sense? Quite resonating?
Yes, so basically what I wanted to know is that how much of the memorability can be explained by the novelty familiarity axis, and by novelty familiarity, I don't mean just the repetition suppression, because I mean deep familiarity. Like repetition suppression, of course, it can happen on one trial, but by familiarity, I mean long.
I see. You're talking about how much of it can be expressed by atypicality.
Right,
like
like that.
Atypicality with the specific.
Right.
I don't even know how to quantify that. I don't even know how to like. So what I can say is that I think that atypicality is crucial to understanding. So you can ask the question, well, it's fine that deep nets trained to categorize objects, they do this too. It's a little counterintuitive to me because I would have thought like, wouldn't a deep net for the thing that it's like the most typical, isn't that the thing you would think it would like respond the most vigorously to? It's actually the opposite in the deep net. So the fact that the deep nets also have the most vigorous responses to the things that are the most atypical / the most memorable. It's something about the optimizations based on the statistical structure of images and objects. And to date, I have not heard a good theoretical description of how atypicality drives a neural network trained to categorize objects in order to respond. But I think there's definitely a whole thing to go after there in the theoretical world, for sure.
Thank you so much, and thank you for accepting the invitation.
Oh, well, thank you for inviting me. It's great to meet you.
Thank you very much. We have 2 minutes before the end of the session. Maybe we have time for one more question, and then we go to the panel. There's a few questions written. Sahan asked, thanks for such a great presentation. My question is about the rule of 2 parameters on memorability experiments. The interval time between the first and the second time an image is shown to the subject. Are memory decay patterns over time? I'm not sure. Also different in more memorable images related to others. Something like a smoother decay over time.
Yeah, we don't have good data for that in my lab, but Ode Oliva has nice behavioral data for that in that Khosla paper that I cited, where they demonstrate that the decay, you see a decay in the preserves rank order. So there's kind of a decay that preserves rank. They quantify it even. They use it to regress in their equations. So that's where I would point to. And there are other work that's extended that question. So they looked at it over the course of minutes and hours, and others have now extended that into days and weeks and found that memorability decays in the same way on average under these conditions, yeah. But of course, there are going to be all sorts of things that are going to influence that. Attention and different types of memorability, right? So emotional valence is a class of memorability. I suspect that it's supported by different mechanisms than object category, et cetera, et cetera. Yeah, so I think there's a lot to be investigated there.
Okay, great. Thank you again for your time. It was a great talk. And the people that registered for the panel, please join us on the hall, and you can ask more questions from Dr. Ross.
Thank you, everyone. Appreciate it. Take care.